All right, so spinal curves. So if we look at the spinal column of an adult, we have a curve that goes like this at the neck, a curve that goes like this at the thorax, another that goes like this at the lumbar, and then a sacral curvature. All right, so the two primary curvatures are the convex ones, so thoracic and lumbar. Or, I'm sorry, and sacral. So those are the primary ones. The secondary ones are the cervical and the lumbar. So when we're born, here's our head and there's our spine. We have pretty much just one curvature, and that's the thoracic and sacral curvature. After birth, as we start to be able to control our heads and then stand upright, is when we get the lumbar and cervical curvature. So 4 curvatures, 2 primary, 2 secondary. All right. You should be able to tell the different vertebrae apart. We talked about different characteristics of each that allow you to do that. So like in the cervical vertebrae, the biggest thing is they're the smallest. So cervical vertebrae are sort of delicate and small. They have 2 big holes in their transverse processes. That makes them easy to spot. And then their spinous process is split. It makes a kind of V. It's bifid, we call that. So that's the cervical. In the thoracic, we have 45-degree angles. So the transverse processes stick out here at a 45-degree angle. The spinous process is very thin, and it points downward. So it angles inferiorly, we would call that. And then the body is sort of heart-shaped, unlike the body of the cervical, which is sort of oval in the long direction. So that's thoracic. And then in lumbar, these are the largest. Big, heavy vertebral body, so it makes up most of the bone. The transverse processes go directly out to the side. So here they're at 45 degrees, it makes like a Y shape. Here they're straight across, it makes like a T shape. And then the processes are all sort of short and stubby. They're heavy and thick, but they don't extend very far. So be able to tell the vertebrae apart.
Are there going to be there going to be pictures on this exam?
No.
Okay.
No, there aren't pictures. I just made the exam this morning too, so
okay.
There are no pictures on these. All right. Let's see. So tell those apart. Scapula anatomy. The scapula is one of the more complicated bones that we've looked at. And just some key spots I want you to put some things together. Now we've learned about the rotator cuff, right, and the muscles that make it up. So that makes the scapular anatomy make a little bit more sense. All right, so here's the spine of the scapula. So this is the back of it. When you look at somebody's back, you can see the spine. Above the spine is a sort of a flattened area up here called the supraspinous fossa. In that fossa is the supraspinatus. So the supraspinatus is one of the rotator cuff muscles, right? So it lives in the supraspinous fossa. So it lives up here. Underneath the spine in the back, we have the infraspinous fossa. Living in there is the infraspinatus. So the muscle and the spot of bone sort of share a common name. So supraspinatus is up here, infraspinatus is here. And then on the other side of the scapula, we have this broad, flat plane here called the subscapular fossa. What's in there? The subscapularis. So 3 of the muscles of the rotator cuff have fossae, or fossas, on the scapula. The supraspinatus, infraspinatus, and then the subscapularis. So surrounding the scapula are the muscles of the rotator cuff. The only one that isn't in there is the teres minor. And the teres minor comes from the lateral border here, and it heads up to the humerus that way. So review your scapula. All right. Remember, the coxal bones are the 2 big bones of the pelvis. The pelvis is made up of 3 things: 2 coxal bones, and then the sacrum in the back. So the coxal bones are where the acetabulum is, so where the hip socket is, and they're made of 3 bones that have fused together. So the ilium, the ischium, and the pubis. So the ischium makes up those ischial tuberosities, the butt bones, so to speak, where you sit. The ilium makes up the broad crest or shield that sort of sticks up on the sides. And then the pubis makes up the anterior portion of the coxal bones where they come together in the front. So ilium, ischium, and pubis. You should be able to tell the difference between male and female pelvises. So some key differences. The angle between the pubic rami here, in males it's less than 90 degrees. In females it's greater than 100 usually. So it's obtuse angle versus acute angle. The distance between the ischial spines is much smaller in the male than it is in the female. The male pelvis is sort of heart-shaped, and the female pelvis is sort of oval. And then the other thing that's different that you can't really tell in these pictures is the male pelvis is heavier, the bones are heavier, and in the female pelvis, the bones are a little thinner and lighter. So you should be able to tell the difference between those two. Let's see. And that's pretty much the skeleton. So most of you are studying for the skeleton you did in lab. So yes, there are going to be questions about, which bone connects with which bone, or which bone is found in the arm versus the leg, that kind of thing. But you should already know that from lab. All right. Then the next chapter is articulations, and we talked about that there are multiple different types. Now, this is the category you should know here. I don't spend a lot of time learning all of these, but because I'm going to limit it to these three categories. So, synarthroses have no movement, so they're a joint because two bones have come together there, but it doesn't move. So that's the synarthrosis. Those are things like the synostosis, the sutures, the teeth in the jaw. So these are our bone connections that don't move. The amphiarthrosis is a little bit of movement. So it'll flex or bend, but it certainly doesn't move freely. And then the last one is the diarthrosis. These are the synovial joints. So these are joints where the bones can freely move with respect to each other. So synarthrosis, no movement. Amphiarthrosis, a little. And diarthrosis is another name for synovial joint. All right. We spent the first part of that chapter talking about common features in joints. So the articular cartilage, remember, covers the ends of the bones. It's slick and smooth, and it allows the bones to very smoothly rub across each other or move next to each other. The synovial fluid is a thick kind of oily fluid that's found inside the joint. It keeps everything nice and lubricated. The fat pads are adipose tissue that sort of packs in and around a joint to allow it to move, but also be protected on the outside. Bursa are fluid-filled sacs that allow friction to occur without, allows things to slide across each other, basically. Ligaments connect bone to bone. And then accessory cartilages are things like the menisci in the knee, where they're cartilages that are not directly attached to the bone, but help to keep the bones in their proper position. So we have some of those. We talked about different joint types. Where are we? All right. So we have a gliding joint. This is where the bones sort of can move with respect to each other. The hinge joints, which we see in the elbow and the knee, and this is where bones can move but really in only one plane. A pivot joint, like we see in the atlas axis, a pivot just means to go around. So we see that at the head, and we also see it where the radius and ulna connect as that radius spins. And then we have ellipsoid joints, saddle joints, and ball and socket joints. So particularly pay attention to the ball and socket the hinge, and the pivot, because those are ones that you may see again. All right. Let's see. All right, motion words. Okay, these are critical. There are a number of questions about these words on the test because I told you that these are some of the most important words to learn in the course. So be sure you know all of these. So flexion and extension at every joint. You know, that's not hard to do. You have a wrist, you have an elbow, you have a shoulder. So think flexion, extension at each joint. You have hyperextension is where it's extension past anatomic position. You know, so like for the shoulder, that's extension, that's hyperextension if you put your hand behind you. Abduction and adduction. Abduction is to take away, so to spread the arms out. Adduction or adduction is to bring them back in. At the hand, it's spreading the fingers is abduction and pulling them in is adduction. The wrist can do the same thing. This is circumduction, drawing a circle in the air. It's a ball and socket trick. So we see that at the shoulder and at the hip. And then rotation. The head rotates. The trunk also rotates. You can rotate your trunk. And then for the arm, you have supination and pronation, as well as rotation of the humerus. So external rotation and internal rotation. We see this at the hip as well. All right. And then the special movements, which I'm not going to go over again, but certain areas of the body get special words, you know, like the foot with its dorsiflexion and plantar flexion. And then I suppose the jaw, depression and elevation are key ones. So when you chew, you elevate and depress repeatedly to match the food up. All right. Motion words. Okay, and then some specific joints. All right. So at the vertebral column, we have this stack of vertebral bodies. Each of them has a disc in between called the intervertebral disc. That disc has two parts. Around the outside is the annulus fibrosus, which is sort of tough and fibrous. And then on the inside is the nucleus pulposus, which is kind of gel-like. So it's shock absorption. Running the length of the vertebral column are a number of ligaments that we talked about. So along the front side is the anterior longitudinal ligament that connects all the vertebral columns. Along the backmost part, at the tips of the spinous processes, are the supraspinous ligament. So that's up here. In between the spinous processes are the interspinous ligaments. So there's one, there'd be another one here. Now inside the vertebral canal, the front of the vertebral canal has a posterior longitudinal ligament, so it runs the length this way. And then the back of the vertebral canal has the ligamentum flavum, which is one of the stretchiest ligaments in the body. So the back of the vertebral canal, which is right here, is made up of the ligamentum flavum, and the front is the posterior longitudinal ligament. All right, so the ligaments of the vertebral column. All right. The elbow. The elbow is a unique joint in the body because 3 bones come together at the elbow. At the knee, it's only 2 bones. Even though there's the fibula is down there, the fibula doesn't actually connect with the femur. So at the knee, you only have the tibia connecting to the femur. At the elbow, you have 3 bones that all connect. The humerus, the radius, and the ulna all come together at what is called the elbow. So it's a joint that has 3 bones, 3 articulations, essentially. The ligaments that we talked about, the annular ligament surrounds the head of the radius and allows that radius to spin in there. Annulus just means lens-like, which means it's round, which it is. And then the ulnar and radial collateral ligaments are found on the side of the elbow.
holding the bones in place. So on the lateral side is the radial, and then on the medial side is the ulnar collateral ligaments. All right, so 3 bones come together there. The strong part of the elbow joint is the ulna humerus part. So the humero-ulnar joint is the strongest joint at the elbow. And then the radio-ulnar joint is the most flexible because it does that spinning motion that we saw a video of. All right. So that's the elbow. And then the knee. The knee is femur to tibia. Because you can see that the fibula is way down here, nowhere close to the femur. So the knee joint is just 2 bones, femur and tibia. This is the end of the femur that we're looking at here. So there's a medial condyle and a lateral condyle. This is the head of the tibia. And then between the femur and the tibia are those menisci, which are sort of lens-shaped in that they're convex. And these rounded condyles sort of fit in those menisci. And the menisci help to keep the femur and tibia lined up right. Okay? And then there are a number of ligaments that you got to know. So you have 2 cruciate ligaments on the inside of the joint. So the anterior cruciate ligament and the posterior cruciate ligament. And then on the side of the joint, you have collateral ligaments that hold the bones in place. Now this is where we have some unfortunate naming. Your book will call the lateral one the fibular collateral ligament. Everybody else in the world today would call it the LCL, or the lateral collateral ligament. So you kind of have to know both names. On the medial side, your book calls it the tibial collateral ligament, but everybody else in the world calls it the medial collateral ligament, or the MCL. So MCL, LCL, and then the ACL and PCL, and then the menisci in between. All right, so the knee. The knee and the elbow are really the only 2 joints that I asked you to know very much about. So know those ligaments from the elbow, know the ligaments from the knee, and you should be in good shape in terms of joint specifics. All right. And then we get into, sorry, all lights are off. We got into that skeletal muscle tissue chapter, which is all physiology. And we're going to catch another chapter like that when we get to the nervous system and we start talking about how nerves work. So we're going to get into some of that complexity there too. But first up, muscle is organized as bundles of bundles. Oh, not that one. All right, so there's a bundle of proteins that's called a myofibril. Myofibrils are repeating sarcomeres. So remember, a sarcomere is an arrangement of thick and thin filaments, and they sort of chain together. Like here's a sarcomere, and then here would be another one over here, one after another like this, into bundles called myofibrils. So a myofibril is a long string of sarcomeres. There are many myofibrils in a single muscle fiber. A muscle fiber is a muscle cell, same thing. And then there are many muscle fibers in a muscle fascicle. Remember, a fascicle is a bundle of muscle fibers. And then a muscle is a bundle of fascicles. So we have bundles of bundles of bundles. So from the smallest is the myofibril out to the largest, which is the muscle itself. All right. The sarcomere itself, it has thin filaments and thick filaments. The thick filaments are bound together to each other at the M line. So the M line is here. The thin filaments are bound to each other with the Z line here. And then we have a number of bands. So the I band is the area where there's only thin filaments. The A band is where there are thick and thin filaments present. So there. And then the H band is where there are only thick filaments. So do yourself a favor and review the naming of the sarcomere, and you'll get some easy points, I think, because there is a couple of questions about the sarcomere on there. Not because I'm being nitpicky, but because it really is the fundamental functional unit of the muscle. So you have to know a little bit about the sarcomere. All right. And back. Oops, too far. All right. Then thin filaments and thick filaments. Remember, in order for muscle to contract, actin and myosin have to interact. So the myosin can pull on the actin and make that muscle contract. So we divide the components of the sarcomere up into thin filaments and thick filaments. The thin filaments are the complex half, because the thin filaments are where things have to change, where things have to happen. So thin filaments are made up of actin, and actin is a simple coiled helix of little ball-shaped proteins like this. Surrounding that helix is two other helices of tropomyosin. So tropomyosin is also thread-like, and it binds to troponin, and troponin holds it in just the right position to block the active sites on actin. So when there's no calcium around, troponin holds tropomyosin over the active sites on actin, and it prevents muscle contraction. Okay? Now, when we add calcium back in, we'll see that that troponin is going to move, and the tropomyosin is going to twist, and then the active sites will be exposed, and exciting things will happen. So thin filaments are actin, troponin, and tropomyosin. So there are three components in the thin filaments. The thick filaments are where the energy for muscle contraction comes from, but they're much simpler in structure. There's only one thing in thick filaments, and that's myosin. So myosin is this golf club-shaped molecule, and all the handles are sort of tied together into a thick filament that has myosin heads at both ends. So thin filaments and thick filaments. All right. So in order for muscle contraction to work, we need muscle to contract only when it's given the signal to do so. So we had that whole excitation-contraction coupling business, and here's the short version. So an action potential comes down from the brain through a motor neuron, right? At the neuromuscular junction, that triggers the release of acetylcholine. That acetylcholine creates an action potential now on the muscle cell membrane, the sarcolemma. That action potential spreads across the muscle cell. It goes down those T-tubules, and as it passes the sarcoplasmic reticulum, it triggers calcium release from the sarcoplasmic reticulum. So the action potential brings about calcium release. Okay? That calcium release then affects troponin. So calcium binds to troponin. Troponin moves tropomyosin, exposing the binding sites on actin. And then now cross bridges can form. So actin and myosin interact, and myosin pulls on the actin thread and effectively shortens that sarcomere, shortening the whole myofibril and therefore the whole muscle. After that, we get into the, where are we? Is it this one? Yes. We get into the contraction cycle. So once the active sites are exposed here, as long as there's ATP around, myosin is going to attach to the actin. It's going to pull that actin. Then it's going to reset using ATP and then repeat. So we'll go like this, and the sarcomere will continue to shorten until there's no longer any calcium around, and troponin moves tropomyosin back into its blocking position. So a long cause and effect chain we talked about for muscle contraction. You know, this happens, which makes this happen, which makes this happen, and so on. So if you haven't gone through that in a while, go through the steps. How does a brain signal become a shortening muscle? Because that's a big component of what that whole chapter is about. All right. All right, we're getting there. All right, in order for Contraction will continue as long as calcium is around, and ATP. Now, if either one of those things run out, then relaxation is going to occur. So the way a muscle relaxes is calcium is pumped back into the sarcoplasmic reticulum, and then that troponin moves tropomyosin, and then contraction stops. Now, if we zoom out from the inside of the muscle fiber and we look at muscles as a whole, we talked about two primary ways that the brain can vary the amount of tension developed in a muscle. One is with twitch or wave summation. And what I mean here is the more rapidly a muscle is stimulated, the more tension it will develop. You know, if you stimulate a muscle once, it'll just twitch. You know, it'll contract and relax. That is not how our body uses muscles. Instead, the brain sends a signal like that, that. And with each signal, a little more contraction occurs until that muscle is maximally contracted. So that's one way the brain can, you know, lift a feather or lift a bowling ball, is by changing the rapidness of the twitches. The other is motor unit recruitment. So a motor unit is all of the muscle fibers innervated by a single motor neuron. So like here in this example muscle, all of these fibers have 3 neurons that are innervating them. So the purple ones are innervated by the purple neuron, the blue ones by the blue, the red ones by the red. So a key component here is every muscle fiber has only one neuron that innervates it. But a single neuron can innervate multiple muscle fibers. And all of the muscle fibers innervated by a single motor neuron, we call that a motor unit. And the reason that motor units are important is the brain can effectively turn on or off different parts of a muscle at any given time. So let's say you needed to move something that was very lightweight, you know, like a coffee cup. You know, your brain is going to just use a few motor units in your bicep to do that. Now, if you had to go move a couch, now many, many more motor units are going to be used in that same muscle. So this is another way that the brain can control how much force a muscle generates, is by determining how many motor units are going to be participating in that action. The other thing that motor units can do is they can hand off. They can do like a relay. If you're holding something heavy for a long time, wouldn't it be nice if part of the muscle could rest while the other part was working? So we see that relay effect as well when there's a long sustained contraction. All right, so motor units. We talked about the different contraction types. So the typical muscle contraction you think of is called a concentric isotonic contraction. This is where a muscle shortens under a load. So, you know, you're picking something up, the muscle is shortening, the load is overcome. That's a typical muscle contraction. In eccentric isotonic contraction, the muscle is lengthening, but also under load. The easiest example of this is you're setting down something heavy. You know, you don't just drop the couch. You want to set it down so it doesn't go through the floor. So you lengthen your muscles while you're maintaining that force to gradually set it down. And then the last one is isometric. A muscle can contract and create force without shortening. You know, if anybody try to stand on one foot, let's say, for 10 minutes, what you'll find is that your muscle gets tired. Why? Because it's maintaining force even though it's not changing its size, even though there's no motion there. All right. So muscles are energy hogs. Because of that, they have some adaptations for their high energy demands. We talked about creatine phosphate is a way of kind of storing ATP up. You know, creatine gets a phosphate added, and then when the muscle is busy, that phosphate is shuffled back to an ATP so that new ATP doesn't have to be made. Myoglobin is keeps oxygen around inside a muscle, so it's like an oxygen reservoir. And then glycogen or starch, there's really two places in the body you find it. One is in the liver, where the liver stores glucose for the whole body. And then in skeletal muscle, glycogen is stored there for use in anaerobic metabolism. So when ATP needs to be made in the absence of oxygen. All right. So muscle can use energy in more than one way, depending on how busy it is. So resting muscle is going to use fatty acids as its primary fuel supply, and it's going to feed those fatty acids into the mitochondria to produce ATP. Now, when that muscle starts to get used, as long as the oxygen supply can keep up, then fatty acids will continue to be used as primary fuel because they're very energy dense. However, if the amount of muscle activity exceeds the amount of oxygen that can be supplied, then we get into anaerobic metabolism. So this is aerobic over here, where we see the mitochondria being used. This is anaerobic. See how there's no mitochondria there? Instead, it's all glycolysis. So glycogen is broken down to glucose. Glucose is broken down to pyruvic acid. That creates ATP. That's a good thing. That's why we do this. The downside is lactic acid is also produced. So this lactic acid builds up in the muscle. It builds up in the body as well as it leaks out of the muscle. And eventually, when the lactic acid is high enough, the muscle will stop contracting because the pH will be so high. So aerobic and anaerobic metabolism. Now, after there has been a period of anaerobic activity, the body's lactic acid is also elevated. So in order to bring that level back down, we have a system called the Cori cycle. In the liver, lactate, or lactic acid, is built back into glucose. That glucose is released and then restored in the muscle again as glycogen. So the skeletal muscle liver cycle that gets rid of that lactic acid in exchange for glucose, we call that the Cori cycle. And it's an important aspect of recovery after exercise. You know, when you stop running, for example, your heart rate and respiratory rate don't immediately go back to normal. And the reason for that is you have to build the reserves back up. So that oxygen debt, the Cori cycle, is part of why that happens. All right, we're almost there. Ignore that. We're not there yet. Okay, heroic Cori cycle. Okay, and then the muscular system. A lot of questions on what is covered in this chapter. Is it chapter 10? The last one we did? Okay, so here it is, and we're going to talk about this in lab 2. Know about the different fiber organizations. So parallel, convergent, pennate sphincters. Parallel is the most common. Convergent is probably the most flexible, in other words, the most motions that can be made. And then pennate are the strongest. So they each have those differences. We talked just a little bit about the 3 types of levers. So a first-class lever is like a seesaw. Second class is like a wheelbarrow. A crowbar is also a second-class lever. You know, when you go to lift something heavy with a long stick, same concept. And then the third-class lever is the spoon catapult, where you make a small motion at one end equal a large motion at the other. All right. Origins and insertions. The origin is the part of the muscle that doesn't move. The insertion is the part that does. So one end of a muscle is usually stationary. The other end is pulling on a bone. So the stationary end is the origin. The pulling side is the insertion. Agonist, antagonist, synergist. Just like in English class, when you learned about these in literature, same meanings. You know, an agonist is the prime mover. An antagonist works opposite it. And then a synergist is a helper. All right. The carpal tunnel is found in the wrist, on the flexor side of the wrist, and it contains many tendons heading to the fingers. Also in the carpal tunnel is the median nerve. So together those things cause trouble because they're all trying to pass through the same space, so they can irritate each other. And when they get irritated, you get symptoms, both nerve symptoms and muscle symptoms. So that's carpal tunnel. And then the muscles. Okay, most of the muscles that you're going to learn or we're going to do in lab, but the a subset of those, which I think of as muscles that you've probably already heard of and are simple and large and impressive muscles. So here is sort of the list. These are the muscles that have videos attached. If you were in class that day, I said the muscles with videos are fair game. Now, what can I ask you about these? I can ask you where they are. I can ask you what they do. That's it. So I'm not going to ask you origins and insertions or innervations or any of those things. So the short list here is the erector spinae muscles. Remember, I had you add those to your muscle study guide. The external abdominal oblique and internal oblique, and the rectus abdominis, so the abdominal muscles we talked about. The trunk muscles that have to do with the arms, so trapezius, pectoralis, the lats, and the deltoid. The rotator cuff, the sits muscles. Biceps, triceps, brachioradialis, and then gluteus maximus, iliacus, psoas, quadriceps, hamstrings. So the large muscles. And I'm going to give you this list again in lab too, because today we're going to study those muscles so that you'll have seen them by Wednesday, essentially, or have looked at them again by that time. Okay, that was a lot. All right. So let's see. Let's do a question or two while we still have time. So if you haven't gotten logged in, please do that. Which not very many of you have. So all right, I'll give you a minute to log in. While we're doing that, anything in particular anybody want me to cover that I didn't already? Yeah.
I remember in the homework for like the muscles, there was one that had like 10 to 20 cycles or 20 to 25 cycles, and I don't remember us talking about that directly in class. Let me see if I can pull it up.
Yeah, I'm not sure what you're talking about.
I think that was chapter 9 homework, yeah.
Does someone have it handy?
10 to 20
Oh,
cycles?
it's like the 10 to 15 or the 20 to 25.
Yeah.
Which would be more, which works better if muscle receives 10 to 20 like signals or 20
Here, I can pull it up right here.
Okay. Yeah, we didn't talk about it in that detail, but the faster you stimulate a muscle, the more contraction it develops. So that the 20 to 25 would develop more tension than the 10 to 15. But yeah, we didn't get into that level of detail about it. The other thing that I dropped out of the test was the different muscle fiber types. You know, there's slow twitch and fast twitch and medium twitch. You can skip that one because there aren't any questions about that on there. Because I can never keep that stuff straight either. So
the higher the action potential, the more contraction it has?
yes,
Okay.
yes, the faster the cycle. Yeah.
Okay.
Okay, let us find a question. Okay, ligaments. What do ligaments do?
I don't think I put my name in.
It's all right. Kind of a redundant system for that. All right, I guess that's everybody. All right, so a ligament attaches bone to bone. So that's B. Not to be confused with tendons. Tendons connect muscle to bone. So C is a tendon, B is a bone. Muscle to muscle, there's really no such thing. Just like nerve to bone. Yeah. So ligaments are bone to bone. Okay. Which of the following bones is part of the axial skeleton? Last couple of people jump in there. All right, so axial, appendicular, that's how we divided up the skeleton. So the rib is part of the axial skeleton. The rest of those are part of the appendicular. So the radius, obviously found in the forearm. The clavicle, sort of found in the chest, but it's still part of the appendicular skeleton because it's part of the pectoral girdle. So the pectoral girdle, or the arm to trunk connection, is the clavicle and the scapula. So two bones make up the pectoral girdle. And then E, the coxa, or the coxal bones, make up the pelvic girdle. So even though we looked at the pelvis, you know, the two coxal bones and the sacrum in the back, the only part of that that is axial is the sacrum. The coxal bones are part of the appendicular skeleton too. All right. All right, so this is a good example question because there is at least one like this. The sagittal suture is located between which bones? I'm trying to send it to you. And you may not have thought about the sutures like this before. So in the next couple of days, go through each suture and think about which bones is it made from. All right, good. The answer there is A, the two parietal bones. All right, so let's go through the rest of them. The frontal and parietal bones, the suture that's there, that's the coronal suture. That's this one. Between the parietal and occipital bones, that's in the back, that's that lambdoid suture. And between the frontal and temporal bones and the parietal and temporal bones, that's the squamous suture. All right. The cheekbone is also known as which of those things? Aha, you all know that one. That's B, the zygomatic bone. So that's the cheekbone. The foramen magnum is a large opening in the base of the skull. For which of those things? All right, I think that's everybody. And that's for E, the spinal cord. And the foramen magnum is found in the occipital bone. So it's the occipital bone comes down and around, and it makes the base of the skull. So your head sits on your vertebral column on the occipital bone, and in the center of that bone is this large hole that the spinal cord passes through. All right. Hold on. There we go. Synaptic vesicles in the neuromuscular junction contain which of those things? we got kind of a split going on this one. Jump in there if you haven't already. There we go, that's everybody. Okay. So at the neuromuscular junction, first of all, what is that? That's where the motor neuron connects to the muscle. So that's where the nervous system and muscular system meet, is at the neuromuscular junction. The neurotransmitter that's used there is acetylcholine. So what you find inside the synaptic vesicles are acetylcholine. When that action potential comes down from the brain, that triggers calcium to enter the nerve, and then that calcium triggers the release of acetylcholine into that synaptic cleft. Then that acetylcholine is what triggers an action potential in the muscle that starts that whole excitation, contraction, coupling thing that we talked about before. Now, D, acetylcholinesterase, is found in the cleft, but it's not found in the vesicles. And what acetylcholinesterase does is it destroys acetylcholine so its effects are transient. In other words, so that at that neuromuscular junction, the signal can be sent and then stopped. The acetylcholinesterase stops the signal. All right, thank you, everybody. We'll call it a day right there. I will see you, some of you in lab today, some of you tomorrow morning.
Are you going to post it on Angel?
Huh? Yes, I'll put this on Angel, yeah.